Hello, this is Dr. Dan Guerra from VeraMed Studios from the inland northwest of the United States. Today we're going to attack a second aspect of what I had been discussing on the last two lectures. That is, we're going to be looking at what I have invented or described as the diaventome. What that entails will be fully described in multiple subsequent lectures. But if you want to have a detail about the theory itself, or that is the hypothesis leading to a theory, please refer to the last two VeraMed videos I generated that are now on YouTube. Okay, so today we're going to actually look at a specific manuscript that's going to examine how events in the brain that relate to dietary intake, the normal hormonal response for satiety, and how that is mediated through the epigenetic programming of the immune response that helps control dietary influx, as well as indirectly affecting the immune response. So this is all linked together in this grid, which examines how the genome and the environment help generate the immunoepigenome, and working in concert control aspects of the central nervous system and the immune system that relate to not only learning, that is actual learning through synaptic connections, but also the constant turnover and interaction of metabolism that happens offline or that is unconsciously. So again, we're trying to develop this kind of global theory. So let's take a look at this paper. All right, so we're going to get started here from the beginning, of course. All right, so that's me, Dr. Dan Guerra, out in the woods, and this little vignette over here was taken just last week at the St. Joe Clearwater National Forest. No, I didn't cut down all of those trees. You can tell I didn't, because they're all like laying there rotten after the winter where all the snow was on top of them. But I did drop this tree right here, which was a white spruce. Very common in this part of the woods, my neck of the woods. And I want to show you that that tree was dropped here, and then the rest of it took all that wood and put it in the back of cut it all up, put it in the back of my pickup truck. And there's the top of the tree down there. So that's where I dropped the tree down that line. Kind of cool, right? This was actually the result, all these dead trees laying here was the result not of some vast tsunami or some kind of major climactic event. This was actually due to clear cutting that was carried out last year in my forest here. So that's where this picture was taken. That's the road outside it. Okay, enough of that. Anyway, this is VeraMed from VeraMed Studios. There's my Facebook account. You can get all my videos there. You can get to our website directly. You can email us there. You can go to the website and see what we offer in terms of consultation and biomedicine. You can also email me directly and talk about any of the video lectures you've seen or to set up an appointment to start to work through VeraMed. Again, Facebook is really a good place to find or discover all of these videos. Today's the 23rd of April, 2018, and I'm going to talk again today about endocrine-mediated neuronal control over T cell epigenetic reprogramming. I know that's kind of a lot of complexity in the title, but I will unravel it hopefully sufficiently that all of it will be clear by the end. All right. Now, first let's take a look at what paper we're going to examine. Paper published in the Proceedings of the National Academy of Sciences on April 9th, some 5 years ago in 2013. There is the reference. So what are we going to be talking about today is going to be embedded in the introduction of that paper, and I've rewritten it here. Hypothalamic neurons that express the orexigenic agouti-related peptide, that's going to be called AGRP, are necessary for endocrine control over the feeding response. So don't worry about the agouti-related peptide. We're not going to talk about it today, but that's a way to describe a specific kind of neuron, okay, in the hypothalamus. So there are various nuclei in the hypothalamus, and those nuclei have certain neurons, and one class of neurons are the AGRPs, okay, because they express that protein. Now, orexigenic means that it's related to feeding intake, right? And so what we're going to be talking about are endocrine hormones that are sent to that part of the hypothalamus that are going to regulate satiety or the feeding response. The one in particular we're going to look at is leptin, which comes actually from the adipose fat or the adipose tissue. But we'll get into that. Now, these same neurons, these AARPs, express not only all of the regulation that we're going to be looking at, but they also express a histone deacetylase, sirtuin 1. Now, you will recall from various talks I've given in the past, sirtuins are really one of my favorite new enzymes. And I love a lot of enzymes because I've been enjoying enzymes for years and years and years, in fact, decades, to be honest. Sirtuin is one of a family of deacetylases which remove the acetate molecule from various substrates, including things like histones, which are those proteins that cohere with DNA and chromatin. And histones, when they cohere with DNA and chromatin in eukaryotic cells, help control the condensation of that DNA and both directly and indirectly relate to how that DNA can be transcribed, that's how you get gene expression, and also how you can recombine within the genome and repair. So histones are very important in the dynamics, the topodynamics of DNA metabolism. So when you acetylate a histone, you alter that. When you deacetylate, you bring it back to where it was. Adding that acetate to those histones actually opens up the DNA, and removing that acetate, which is what our sirtuins do, collapses that DNA and generates a structure that used to be called heterochromatin, which limits the total scope of genes that can be expressed because it limits the ability for RNA polymerase and all those other proteins involved in transcription to be able to enter into the chromatin in such a way that you get remodeling, and that remodeling then resulting in gene expression. Okay. And I talked about sirtuins many times, so please refer to those or just go ahead and just Google sirtuins and you'll find out a lot about them that I haven't already said today. Now, so you're going to talk about sirtuins too. So now let's go on. Behavioral and metabolic adaptations to low energy availability. Again, something we talk about a lot in Vera Med, as detected via the concentration of ATP, and more importantly, the AMP kinase, which is on the other end of the spectrum, are obtained via the hypothalamic AARP neurons. Okay, so what we're saying is that metabolism is going to be integrated into this feeding response, which makes sense, right? So if you have a low energy budget, what does the brain need to know? The brain needs to know that you need to take in calories, right? But when those calories are taken in, the brain likewise needs to know, and it's working through the hypothalamus here, that feeding should stop. And so that's what we're talking about here. We're talking about hormonal control over the feeding response, okay, at first measure. And then we're going to be looking at how these deacetylases help pattern that response and ultimately how that's going to affect the periphery and the immune response. Yeah, really cool. All right. And it's a behavioral response, right? When you feel full and when you stop eating, that's a good thing. Right? When you're hungry, you consume food, right? So it's going to have actually not only an endophenotypic effect on synaptic control of action potentials, it's also going to have a direct effect on how you behave, whether or not you're going to eat more or not. So that has to do with things like thought processes. All right, so sirtuins are, again, NAD-dependent class 3 deacetylases, just a type of enzyme they are. And caloric restriction, okay, so when you stop eating or fast, upregulates sirtuin 1 expression in several tissues. So that's just something that's given in the literature. Now, SIRT1 activity is linked to metabolic shifts that occur during negative energy balance. That means when you have when you have a deficit of energetic stores in the form of, for example, classically ATP. And that balance, which compromises a metabolic state in which energy utilization is higher than energy production, obviously. Okay. Now, thus, sirtuins are placed perfectly because they mediate the beneficial effects of caloric restriction. So this is one of the reasons people are interested in sirtuins, because maybe if we can find a way to signal through them, we might be able to inhibit the feeding response and get at one of many ways at getting on the obesity epidemic. See? There you go. All right. Now, you'll recall from the last two lectures that I introduced my concept of the diaventome. Now, I'm not going to go through the detail of that now. I'm just going to tell you that I claimed there is a trigonal planar connection that involves the endocrine system, the nervous system, and the immune network. Okay? That's where you get the trigonal or three-component or triangular, right? Although the central nervous system was initially considered to be immune privileged, way up until really the 70s and into the 80s, with a lack of immunosurveillance, it has now been firmly established by scientific evidence that there is a robust immune network functioning in the CNS, albeit its function kind of still really misunderstood. What's it doing there? Probably not involved in getting rid of pathogens, right? Because you don't have a whole lot of bacteria or virus or fungi in the brain. Okay, so that's not the case. So what is it doing there, right? All right, that's what we're going to talk about. Now, the autonomic nervous system. Okay, so back up here. The central nervous system influences and controls the immune system via the autonomic nervous system. So it's going to have to be some description, some recapping for those of you that have had all this in medical school or as graduate students or maybe just as biology students. I'm going to have to recap a little about the ANS and the SNS and whatnot. Don't worry, we'll cover it. It's not complicated. Anyways, the ANS, autonomic nervous system, is separated into the sympathetic and the parasympathetic. See them as one working against the other in a way, right? Okay. Lymphocytes, remember what those are. Those are immune cells, right? Those are things like T cells and B cells. Express variable neurotransmitter receptors on their surface. Thus, they can coordinate the immune system via neurotransmission. Isn't that interesting? Okay, when you start learning about that immune cells have receptors for neurotransmitters, now you're starting to see how things come together, how that triangulation comes together. All right. These are just observations based on the evidence of looking at the literature. Now, indeed, the nutritional status can affect T helper cells, and they are involved in a pro-inflammatory immune response through a series of adipose tissue-derived hormones. Now, there you go. We're starting to put things together. Nutritional status, that would be the environment, can affect T cells, particularly the class that are pro-inflammatory, and that works through a hormone that comes from the adipose tissue. In this particular case, the hormone known as leptin, the orexigenic hormone, which, don't worry, we'll talk about that in a moment too. Now, leptin, in fact, right here, directly activates leptin receptors. Gee, that's really interesting and bizarre. On T cells, though, and it coordinates this with the autonomic nervous system. There you go. And now you've got not just neurotransmitters, now you've got an endocrine hormone that has a receptor on T cells. You see how this is starting to really triangulate, right? You're starting to see how diet, the immune system, and the brain are working together, relating to a behavioral response such as the feeding, right? Whether or not you need to eat or not. All right, now take a look at a paper that was published in Molecular Metabolism, one of my more favorite journals, newer journals these days. Came out just recently in 2018. Okay, let's take a look at this. Energy homeostasis is, of course, controlled by the central nervous system. Your brain is the thing functioning that controls energy homeostasis. I don't think that's anybody's surprise. Particularly, the hypothalamus and the brainstem receive signals on energy and nutritional status transmitted from the periphery. For example, hormones like leptin, insulin, which we all know comes from the pancreas, ghrelin, which comes from the stomach, thyroid hormones from the thyroid gland, and the gonadal steroids. Those are just some of the ways that the brain responds with hormones. Okay, it's a lot, right? Information from those signals is integrated in the central nervous system and modulates different aspects of energy balance, such as food intake, energy expenditure, or control is mainly exerted by two complementary and non-exclusive drivers. They are the endocrine system and the autonomic nervous system. Now, the ANS innervates, several peripheral organs or tissues, including brown and white adipose tissue called BAT and WAT, bad acronyms, liver, pancreas, gut, kidney, adrenal gland, and skeletal muscles. So it's all over, right? The autonomic nervous system innervates all of these systems. But maybe you probably knew that it innervated things like the liver and pancreas, and probably the muscle, and an adrenal gland, an exocrine gland like that. But you probably didn't think about the fact that the ANS actually innervates adipose tissue, because the adipose tissue is basically another, it's a structure much like an organ in the body. In fact, it's very important for energy homeostasis in humans. Now, the ANS consists of the sympathetic nervous system and the parasympathetic nervous system, where the SNS is associated with catabolic responses and the PSNS with anabolic responses. Catabolic breakdown, anabolic buildup. So therefore, you can think about energy intake and energy utilization, energy sequestration, and an energy requirement, all within the SNS and PSNS. Now, under some physiological states, both SNS and PSNS, which are part of the ANS, can be activated or inhibited at the same time. But typically when one is activated, the other is inhibited. Now, I want you to understand there's not an exclusivity here, okay? There are parallel systems. The SNS and PSNS are not one's on and the other's off and vice versa. It doesn't work that way. So they're not contradictory, right? They can be contrary in a way, but they are not contradictory, right? Thinking about the square of opposition there, right? All right. Now, again from this paper from Molecular Metabolism, leptin, which is this peptide hormone from the adipose, modulates the ANS via a particular type of neuron called the POMC and the neuron we've already examined, the agouti-related peptide neurons. You got two different ones. POMC stands for pro-opioid melanocortin. Okay, now that we also discussed in various previous VeraMed lectures. And that's a really complex gene that when it's expressed as an RNA will generate all kinds of interesting proteins. We're not going to talk about them today. But some of them, of course, are the endorphins and enkephalins, right? The naturally occurring morphine-like molecules in the brain, right? Okay. Now, anyways, that's all I'm going to say about POMC. We're not going to talk about them again today, but keep that in mind. That's in the hypothalamic arcuate nucleus, and that leptin is going to modulate various organs and tissues, okay? So you're going to see it right here. The modulation affects both the SNS and the PSNS, innervating, as we just said, bat, wat, liver, gut, lumbar, skeletal muscle, and kidney. So, on with the glasses. This is the hypothalamus with all of its nuclei. Okay, it has multiple nuclei, and each of the nuclei have a different function, structure-function relationships. It's going to receive the signal of leptin. Now we're looking at specific neurons, the POMC neuron and the AARP neuron, right? Presynaptic, postsynaptic system, right? It's going to be signaling through here with action potentials. Now, the hypothalamus can work directly on the adrenal gland. That's called the HPA axis. We won't talk about that today. But leptin can also signal through these neurons. And when it does so through the ANS, both the SNS and the PSNS, it's going to affect, because it innervates, brown adipose, white adipose, it's yellow here because that's really what it looks, really the color, I suppose. Liver, the splanchic, the skeletal, that's the gut, you see, stomach and the intestines, skeletal muscle, and the kidney. Okay, now, arcuate neurons evoke those effects by modulating pro-autonomic sympathetic and parasympathetic neurons in all those other nuclei. And so the other nuclei involved are going to be the paraventricular, the dorsomedial, and the ventromedial. Those are all other nuclei in the hypothalamus. You see, those are all hypothalamic. That's what that H stands for, nuclei. All right. So they're not just working through the POMC and the AGRP. All right. Now, notably, leptin-mediated effects on the sympathetic nervous system, innervation of adrenal gland, see there, is independent of those neurons. So we're not talking about that, but it's something to keep in mind. That's your HPA axis. All right, here we go. Here is this slide I got. I don't remember where it was, probably some textbook I used years ago. Scheme explaining how the parasympathetic and sympathetic nervous systems regulate functioning organs. Let's take a look at the parasympathetic. Okay, it's involved in constricting the pupil, salivary glands, slows heartbeat, it affects lungs, it affects the stomach, the liver, the intestines, and the bladder. Okay? Notice where the spinal column is involved there, all that innervation going up and down there. Now, the sympathetic dilates the pupil, whereas the parasympathetic, you see, constricts it. Salivary glands, okay, it inhibits salivation. Heart, it accelerates the heartbeat, the sympathetic does. Lungs, it dilates the bronchi, whereas the parasympathetic constricts the bronchi. See how these look like they're working in contradiction, but they're not really, because remember, it's not on-off. It was on-off, and sometimes you'd be dead, and other times you'd be dead, right? Because if one is only working completely, you wouldn't have a living system. You have to have a mediation, right? And that means both of them are working simultaneously, right? Therefore, they are not contradictory. And on and on and on. You see that one works sort of in opposition to the other, but they're functioning in a commensal way, if you will. All right. Now, here's another great diagram from another journal, which I like to read, European Journal of Endocrinology. Came out about a year ago, and I've used this in previous talks. So take a look at this. Now, we have a Gprotein coupled system, okay? That's what you got here, the alpha, beta, gamma, and there's the AMP kinase right there, okay? So you've got various stimuli that are going to affect the hypothalamus and the hindbrain, okay? So you've got adiponectin, the AARP, that is the protein itself, cannabinoids, ghrelin, glucocorticoids, and even just plain old hypoglycemia, that is low blood sugar, okay? And here you're going to have negative effects by lipoic acid. Don't worry about that. That's a component of a lot of enzymes. Amino acids, various things, CCK, CNTF, citrate, estradiol, the GLP-1, glucagon, glucose, insulin, lactate, leptin negatively affects this system, and vitamin B1. Okay, now when this system is turned on, it works through the calcium-mediated kinase. It, also, you have the LKB, which is another kinase, and this PP2 kinase working in opposite, so it blocks this, right? That's a phosphatase. That's not a kinase, excuse me. The phosphatase that inhibits these two kinases. But when you phosphorylate the threonine on the AMP kinase system itself, right, you do something. First of all, you cause ATP producing processes. You have energy production in terms of food intake, and you shut down ATP consuming processes, right? At the cellular level and the whole body level, energy expenditure, such as thermogenesis, is also shut down. All controlled by all of these factors in the brain, working through this phosphorylation cascade. There's a dephosphorylation pathway, which is inhibited by high levels of AMP to ATP, which turns on the AMP kinase, which then shuts down ATP consuming and turns on ATP producing, okay? So let's take a look. The structure of this AMP kinase is similar to G proteins. It has an alpha, beta, gamma structure, activated via phosphorylation of a threonine, a specific one, on the alpha subunit by those two kinases. It's allosterically activated by AMP. That's why it's an AMP kinase. Induces phosphorylation of threonine 172, which we just saw, but it also inhibits the dephosphorylation, okay, which would then render it inert. Switching off are the red lines, ATP consuming processes. Switching on to the green, or the catabolic process to produce ATP, basically. So the activation of the AMP kinase increases food intake. That's what's needed. That's what's telling the brain. The brain is delivering this message via afferent neurons, explaining to the brain that bioenergetics are south because AMP kinase is turned on. So the activation of the AMP kinase increases food intake and decreases energy expenditure. For example, one type is thermogenesis. So that's what this diagram is showing you, how you link together, right, these various complex signals, which are many of them are just proteins, right? Proteins are peptide factors, or sometimes just plain old hypoglycemia, that are going to mediate a response to this AMP kinase, which we talked about in the past. They're going to shut down ATP consumption and turn on ATP production. All right. Now, why is that important? Well, you'll see. Activation of the AMP kinase increases the appetite, okay? So that's something we're interested in with obesity, right? AMP kinase is highly expressed, again, in the arcuate nucleus, and also in the dorsal medial and the paraventricular and ventromedial nuclei, as well as even in the lateral hypothalamic area, or the LHA. So this is all about the hypothalamus, okay? All those nuclei. The hypothalamic AMP kinase, therefore, is a part of an adaptive change in the physiological regulation of the feeding response, and fasting increases, but refeeding inhibits the AMP kinase activity in the hypothalamus. Okay, so that makes sense, right? Because fasting means you're going to want to increase appetite, right? Eventually. All right. So the hypothalamic AMP kinase dominant negative isoforms decrease expression of the orexigenic neuropeptides, both the agouti-related peptide and neuropeptide Y in the arcuate nucleus. Okay, so that's when you have a double negative. Overexpression of that same gene, AMP kinase, constitutively active, this is the opposite of having a double negative, elevates a fasting-induced expression of those same proteins, the AARP and the NPY in the arcuate nucleus, as well as expression of the melanin concentrating hormone in the LHA, the particular nucleus of the hypothalamus. So in this way, AMP kinase modulates the expression of NPY and the pro-opiomelanocortin gene construct by regulating autophagy. So there's something else here that's going on that has to do with autophagy in the cell. And I talked about this before. So I want you to keep that in mind. This is all going on in the hypothalamus. So let's take a look one more time. AMP kinase senses, okay? AMP kinase is going to sense several nutritional hormonal stimuli. Here they are again, right? There's adiponectin, there's glucagon, there's cannabinoids, right? Working through, there's the AARP, there's neuropeptide Y, right? And what is going to happen is going to mediate specific regulation of the sympathetic and parasympathetic nervous system. So we're putting this back together. The fact that the inhibition of the hypothalamic AMP kinase leads to anorexia and increased thermogenesis and therefore elevated energy expenditure, because that's what would happen if you inhibit that enzyme, makes it, of course, an interesting target for drug development from the pharmaceutical industry, with its potential for controlling both sides of the energy balance equation, okay? Because this is ultimately going to give you body weight control and energy homeostasis by working through the brown fat and the white fat and all the organs involved in regulating bioenergetics, right? All through the hypothalamic nuclei, right? Which is found here in the brain, okay? And we already looked at these individual components and how they work through these given enzymatic complexes. There's the AMP kinase, right? And where we might want to find a way to control AMP kinase would be up here in the hypothalamus. That's what it says here. Anti-obesity drug, question mark, right? All right. Now, that was just the background. Let's go back to the PNAS paper. The evidence suggests that SIRT1, that's the deacetylase, negatively regulates T cell activation, and it plays a major role in clonal T cell energy in mice. Now, clonal means different T cell subtypes. And energy means kind of like putting T cells offline. That's what energy means. This could mean that chromatin deacetylation limits developmental or differentiating gene cluster expression, otherwise necessary for T cell activity. That might mean that that's what SIRT1 is doing, okay? Now, the upregulation of SIRT1 expression led to a T cell anergy, that means putting it offline, by diminishing the activity of the transcription factor activator protein 1 through the deacetylation of a protein called C-JUN. So we deacetylate not just histones, we also deacetylate other proteins, right? And we can acetylate them as well. Now, when that happens, it appears straightforward, but deacetylation begs the question of what is being deacetylated and what is the temporal endurance of that deacetylation. How long is that maintained? That's what I mean by that. That is, what are the protein substrates for deacetylation? Do they work in coordination? And is that signature, the acetylome, penetrant? And does it obtain a sufficient cellular half-life, how long it lasts, temporal, to be more than just a crude correlation? So we've got this SIRT1 activity, and we're saying, wow, this looks really interesting. Looks like acetylation plays some role, but we need to put all this together in real time, and we have to look at real concentrations of intermediates and see whether or not, when you put all that evidence together, you don't just have some kind of crude correlation. You actually have something that looks like they are correlated. That's the point of science too, right? Now, the authors of that paper, 2013 paper, are going to remonstrate the reader, okay, by noting that SIRT1 deficient mice were unable to maintain T cell tolerance. So they're already telling you that Something's unusual here that may throw a monkey wrench into these hypotheses. And developed severe experimental autoimmune encephalomyelitis, or EAE. Okay, so we'll talk more about EAE later, but that is the animal model for MS. As well as spontaneous autoimmunity, not good, and further that treating them with resveratrol, which is an activator of the SIRT1, led to decreased disease symptoms as well as to inflammatory cell infiltration in the CNS. So it looks like if you activate the SIRT1, it could be beneficial there, okay, because of this autoimmune arm of SIRT1 activity. Okay, that has to do with T cells. Finally, knockdown of SIRT1 in the hypothalamic AARP neurons, okay, where we're at now, right now in this paper, alters the central perception of hunger, inducing a series of metabolic and behavioral adaptations that resemble those of satiety, despite reduced food intake and body fat mass, a decrease in that. So think about this, right? If you corrupt those neurons, right, by knocking down the SIRT1 activity, right? So if you knock down the deacetylase, that means you don't remove the acetate, you maintain the acetylation of the system, right? Looks what it does. It looks like it mimics this sort of satiety phenotype, like you're full, right? So this could be, again, a target for the obesity crowd. So in the current study, a selective ablation, okay, of SIRT1 expression in the AARP neurons affected peripheral adaptive immunity. This is the reason I'm talking about this paper. How things that go on in the brain related to endocrine control over feeding are going to affect peripheral immune response, and maybe not in the way that we would have predicted. Certainly not in the way that we might want. All right, here's some data. The AARP SIRT1 knockout mice displayed more immature thymic phenotype. Okay, that means that they haven't quite differentiated, with an increase in the percentage of double negative, that's CD4, CD8 thymocytes in the AARP nucleus in the SIRT1 knockout mice. Okay, so that's what that showed. Now, take a look at the data in particular. mTOR kinase plays a key role, of course, in metabolic control of Treg responsiveness. Okay, remember that's an enzyme involved in controlling metabolism. So the group analyzed S6 phosphorylation, which is a readout of mTOR. Okay, so that's what you're looking at here, the phosphorylation of S6, the protein that is normally functioning upstream on mTOR. Okay, so a measure of mTOR pathway activity is whether or not you get phosphorylation of S6. Now there you got, this is just the blot showing you the phosphorylated form, looking at the knockout mouse model and the wild type. You get less of the phosphate added to the the S6 protein, okay? So that says that you get a lower activity of mTOR pathway when you knock out the SIRT1. You get less mTOR pathway. We just told you that you need the mTOR pathway to control the T regulatory response. So you want to have more Tregs to control the T effector cells, right? Those are the off-on switch for T cells. Now looking over here, this is a proliferation based on BRDU uptake, cells that take up BRDU. That's a normal way just to look at cell proliferation. And here's using an antibody to look at the specific signatures on these cells, so you're able to look at only those cells using flow cytometry. The black bar is wild type, and the grayish bar is the AARP SIRT1 knockout. Now, conversely, this is what this data shows you. Upon in vitro stimulation, and that's what you're doing here, you're stimulating, Treg cells from the AARP SIRT1 knockout mice were not classically anergic. See, they're not anergic. They still look like they're proliferating, right? Like their control littermates, because they showed a reduced energy in vitro upon stimulation. Okay, so this is showing you that just that SIRT1 activity is controlling metabolism in the reg cells, but also controlling proliferation. So proliferation might be okay. See, more immature thymic phenotype. You get more of the immature T cells, but you don't have them differentiating and activating appropriately into the Treg response. All right. So now we're going to analyze peripheral adaptive immune response to the CD4 T cells in immunized mice primed with a complete Freud's angiovis. So now you're just inducing them to have an immune response. Now this is the data here showing you wild type again is the black, and the knockout is in the gray. And these are showing you statistical differences. And whether or not you use the anti-CD3, right, for stimulation. No, yes, no, yes. Okay. Lymph node derived T cells from the AARP Sirtuin knockout mice proliferated more after you stimulate through the TCR, that's the T cell receptor, with the anti-CD3 epsilon and 2C11 monoclonal antibodies. So you're triggering the response than the litter-mate control. So you get more, that shows you statistically different, okay? More T cells are going to proliferate after you induce here in the knockout, okay? And they already know that it's not the Treg that looks like it's doing this, right? Now here is just a bunch of pro-inflammatory cytokines. There's interleukin-6, interleukin-17, interferon gamma, TNF-alpha, okay? And we're looking at, again, the wild type versus the knockout. Okay, there's interleukin-2, another pro-inflammatory cytokine. And we're seeing across the board more pro-inflammatory cytokines. IL-2, IL-6, IL-17, interferon gamma, TNF-alpha, boom, 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 boom. So the AGRP, the agouti-related protein SIRT1, that's the nucleus, that nuclei of the hypothalamus, SIRT1 knockout, that's what we did the knockout, express significantly higher pro-inflammatory cytokines, there they are, compared to the control. You also get the anti-inflammatory IL-4 and IL-5 were lower, see, in the knockout. So those are anti-inflammatory, maybe coming from, again, this is setting up a phenotype of inflammation, right? When you knocked out the hypothalamic SIRT1. You already got the brain now talking to the periphery, right? Because that's where these cells come from. These T cells are coming from the periphery, right? From the lymph node. All right, now, one other interesting thing, look at IL-10. IL-10 is less in the knockout, okay? Now, what does that mean? I had to look this up. I didn't remember. IL-10 is immunoregulatory in the intestinal tract. Yeah. So mutations in that gene, evidence in the literature, are associated with an increased susceptibility, this is amazing, to the autoimmune disease known as rheumatoid arthritis. So look at here, we're having an effect even on the immunoregulatory interleukin. That means that this profound effect of this epigenetic change in the arcuate nucleus, particularly the AGRP nuclei of the hypothalamus, knocking out that SIRT1 has a tremendous effect on the immune response in T cells. Okay. Further, okay, this is just showing you the cell sorters. This is flow cytometry type data. I've done a lot of this, different gating channels and whatnot, generating the much simpler to understand histograms. This is fold induction, okay, on all these axes here for these different, utilizing these different proteins, 25, 54, and 49D, okay, the relative frequency of finding those cells. You see a difference between the wild type and the knockout every time. There's more of all of these in the knockout. Now, what does that say? That says the expression of activation markers such as CD25, 54, and 49D, those are expression markers or activation markers, was significantly higher, the expression, together with their increased induction upon TCR-mediated activation in the knockout mice. That's showing you the induction here, right? Okay, so that's interesting, right? That means that you're turning this system on. You're turning this system on to drive the activation markers for all those T cell subpopulations. Now, molecular analysis of that early activation event, this is an early activation event at the T cell level, revealed enhanced phosphorylation of the ERK, it's a kinase, 1,2, we talked about this in previous discussions, and the S6, as well as concomitantly more pronounced degradation of cyclin-dependent kinase inhibitor, which is called P27-KIP1, in T cells from the knockout mice compared with controls. So every step of the way, you're getting more of this when you look at the knockout and you turn it on, right? That's the CD3 the monoclonal antibody CD3 is turning it on. You get more of all of these systems, right? All right. So that's telling you that in these T cell populations, not in the Tregs, but in the T cell populations that are pro-inflammatory, you're turning them on. That's what all this means. See, your mTOR activity, for example, through the S6, you're turning it on. ERK works the same way. So that means that you're differentially regulating the clonal expression of T cells in the periphery from the lymph nodes of these mice, that you've knocked out this epigenetic marker, this Sirtuin. You've knocked out a particular deacetylase, so you allow for the acetylome in that region of the hypothalamus to be maintained high. That's what this is saying. From the brain to the periphery, turning on pro-inflammatory T cell responses. Okay, continuing. All right. This is fold inhibition of the T effector cells here. Look at here. You see, let me just go through the whole thing. Here's using propranolol. Each of these cases is a way of turning on the system, right? So you have a fold increase here in interleukin-2 expression, right? Right there, showing you that when you induce with propranolol, you get a huge increase as compared to the wild type. Same thing here when you're looking at IL-17, huge increase as compared to non-stimulated versus stimulated. And likewise, even down here when you're using this particular construct, okay? Now, let's see, what does all this data mean? You see a differential change of, again, these particular T cell subpopulations. So, analysis of the peripheral lymph node-derived Treg cell compartments in antigen challenge is what you're doing here. Mice revealed that the AARP Sirtuin knockout mice displayed an impairment in proliferation. Okay, this was accompanied by a reduced activity of the mTOR pathway expressed as S6 phosphorylation, indicating an altered metabolic control of the Treg cells, associated with the reduced capacity to suppress pro-inflammatory cytokines, shown there, in the T effector cells, were not inhibited by the Treg cells. Okay. Basically, what this demonstrates, because now you're having the talk between the Tregs and the T effector cells, what this data demonstrates, okay, further on, this is not just like, again, this loose correlation. This is a really tightly regulated thing with two different populations of T cells that are really going to affect the entire immune response in the periphery, okay? Basically, this data demonstrates that the peripheral Treg cell activity becomes impaired by Sirtuin ablation in the brain. And that implies a neural immunoepigenomic axis. That's what this is. That's what's going on here. All right, continuing with data. Also observed that the uncoupling protein 1 level was significantly elevated in brown adipose tissue, okay, of those knockout animals compared with controls, suggesting that neural control over adipose tissue thermoregulatory energy dissipation that could lead to a reduced obesity phenotype if maintained. So if you get UCP1 level elevated in brown tissue, that means you're going to get basically an uncoupling of ATP metabolism so that you get brown fat thermogenesis. Now, a caveat here, mice have a lot of brown fat. Humans have very, 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 very little, at least adult humans. Infants, a lot more, around the scapula. But adult humans, very little. The point we're making here, though, is in the animal model, when you knock out Sirtuin in that particular nucleus of the hypothalamus, here's a readout asking about uncoupling protein, which is going to give you thermogenesis, which means you're going to burn energy, right, making for decrease in the obesogenic system. It works in the positive way. It reduces the obesity phenotype if you can maintain that. That's what it says, because you're looking at brown adipose tissue. Now, down here, Treg cells from the AARP Sirtuin knockout mice produce significantly lower amounts of regulatory anti-inflammatory cytokines. We've seen this already, IL-10 and TGF-beta, both central cytokines involved in Treg suppressive functions, okay? So that's really important, right? So you can see here that you get basically a much lower amount compared to wild type until you induce, right? And that means of IL-10 and TGF-beta. That means that the Treg cells can't work. They can't work like they're supposed to, okay? So that's really a bad thing. So unless you have some kind of blast immune response, if you're just going along with a healthy animal, you're going to have a greatly deficient ability to immunomodulate, and therefore have Tregs suppress the pro-inflammatory response. All right, continuing. We're almost done here. I think this might be the last data. The Sirtuin knockout mice displayed enhanced delayed type hypersensitivity response and increased EAE, remember that's the model for MS, to the myelin autoantigens, which is what you do to induce it. Now, myelin autoantigen is this MOG. This is a particular peptide of a certain size. When you immunize with that, you basically can induce MS. That's what's doing here, right? Okay. There's your wild type down there. Okay. This is days after introducing this to experimentally generate autoimmune encephalomyelitis. That's what this is. Okay. You're inducing it. And look at how the knockout mouse is much different, much higher level than the wild type, okay? All the way out until after the effect of the antigen starts to wear off, and then they come back together, sort of. So the experimental EAE is an animal model, as I've said, of inflammatory demyelinating disease of the central nervous system. Which one are we talking about? MS. This is the model. So the DTH response, which is correlated with that, to adding that peptide, which is called a priming epitope in terms of how the experiment's done, were significantly enhanced in the knockout mice compared with the litter-mate controls. All right, so you get more of a hypersensitive response, more of the full-blown EAE. So what that says is that changes to the AARP, is the agouti-related peptide neurons in the hypothalamus, is associated with knocked out SIRT1, okay, that's what we did here, right, led to brain inflammation. Yeah, okay. So while we think we're doing okay, right, we're thinking if we can regulate that SIRT1, we might be able to do something with the feeding, right, because working through the AMP kinase pathway, remember all of that, right, through leptin, right, through the AMP kinase, regulating then, therefore, what? The whole process, right? Because the SIRT1 is regulating it from the hypothalamus. Holy smokes, what you end up getting also is a triggering of this demyelinating model disease for MS called EAE in the mouse. That's not good. Let's summarize. What we're saying in this paper, what the authors have at least convinced me of, is that bioenergetics, as monitored via metabolic flux and nutritional status, impacts the peripheral immune system, and that's evidenced by the adipokine-mediated control over inflammation. So that's what the leptin does. For example, leptin, the adipocyte-derived hormone, promotes inflammation, while fasting or chronic caloric restriction inhibits it. So already we're saying, wow, why does leptin do that? It looks like leptin's working somewhere around that Sirtuin axis, you see? And indeed it is. Leptin is secreted from the adipose, and it binds to its receptor in the hypothalamus, as we saw in those previous slides, where it suppresses hunger. That's what it normally does, okay? That's why it's an orexigenic hormone. Leptin is linked to body mass by suppressing appetite, and it's mediated by the hypothalamic receptor. It has its own hypothalamic receptor just for leptin. Leptin binding to its receptor controls all of those kinases, right? Yeah. Impairment of the AGRP neuronal responses to metabolic cues by ablating, that is knocking out SIRT1, resulted in a pro-inflammatory state in which the effector T cell function, or T helper cells, right, is enhanced. Those are going to be the pro-inflammatory clones, while the regulatory T cell activity is diminished. Not only do you uptick pro-inflammatory T cell responses, you down-regulate the Tregs, which normally control the T effector response, by regulating the kinds of cytokines that are produced, which can suppress the effector cells, right? The T effector cells that normally are turning on pro-inflammation. That's not a good scenario. All right. This pro-inflammatory immune phenotype emerges in animals despite the fact that they are leaner and have lower circulating leptin compared to their litter-mate controls. That means that this whole SIRT1 knockout supervened on top of the leptin response. That's what it means. So the data therefore argue for the hypothalamic melanocortin system exerting control over peripheral immune responses independent of the circulating. So it's independent of leptin, okay? Because even when you have this lower circulating leptin, and you shouldn't be having this inflammatory response, you nevertheless get it when Sirtuin is knocked out. Okay. So let me give you a small primer on leptin, which I took from a paper of the annual review. It's an old paper, but these annual review papers are excellent for just giving you general descriptions of leptin, just so you remember what leptin is, as either a layperson or as a doctor. Leptin is produced in proportion to depot fat mass. So you make more the more fat mass you have. Circulating leptin communicates and regulates bioenergetics directly to the central nervous system, from the fat tissue to the brain. Leptin suppresses food intake and promotes energy expenditure. For example, through the AMP kinase, right? Prolonged fasting can be attenuated, so you can attenuate prolonged fasting effects by administering leptin, which falsely would signal to the brain that energy stores are saturated. Because again, you're getting this from adipose tissue saying you are full, right? Adequate leptin levels therefore promote energy expenditure for reproduction, tissue remodeling and growth, the autonomic nervous system and the endocrine network, and the immune system. So leptin is very important in understanding hormonal control, endocrine control. But the lack of leptin signaling due to a mutation of leptin, such as in the OBOB mouse or the leptin receptor, which is the DBDB mouse, results in an increased food intake in combination with reduced energy expenditure, and it's a phenocopy for hypothyroidism, decreased growth, infertility, decreased immune function, in spite of all the peripheral such obesity you get from those mice. So leptin is obviously really important here, but we're telling you that Sirtuin supervenes this system. So the agouti-related peptide-expressing neurons are crucial for hunger and adaptation to low energy availability, where their primary actions on feeding, energy expenditure, and glucose metabolism are mediated by their GABAergic efferents to various hypothalamic and extrahypothalamic sites, such as via the AARPs, which is what we were just going through, okay? That's what these neurons are normally involved in, right? The leptin-mediated effect. So what can we come up with in saying, what are the mechanisms that apparently are evolving in real time to explain the involvement of the immune response to brain-associated endocrine control over feeding? Okay, that's what that statement means up there. We're saying that Sirtuin knockout on the AARP neurons impairs neuronal excitability, and it disinhibits the POMC neurons, leading to decreased feeding and increased energy expenditure. Okay, so that would be something you might want when you want to find targets for obesity. Now, the increase in POMC, opioid melanocortin neuronal activity, remember those are in association with the AARPs, and that you increase the activity by disinhibiting, can lead to elevated sympathetic tone through direct POMC efferents in the thoracic segments of the spinal cord. So that's normally how it functions. Since the immune system is under the sympathetic control, the authors therefore suggest that the immune phenotype, which is altered because of the system, observed in the knockout mouse that we were talking about, was obtained via the disinhibition of those neurons. That's what they think the mechanism is. The mechanism that evolved to explain the involvement of the knockout in the AARP. Now, evidence for this is that the uncoupling protein 1 messenger RNA was elevated in both the brown adipose in the brown adipose tissue of the AARP Sirtuin knockout. Okay, so you remember you get an elevation of the uncoupling protein, you get more brown fat thermogenesis, compared with glutamate controls, which necessarily obtains from elevated sympathetic tone. When you see this means it's an elevation of the sympathetic tone. Okay, so that means it's a direct effect on the sympathetic tone. The hypothesis is that the sympathetic nervous system plays a crucial role in mediating the effect of impaired AARP neuronal activity on the immune system. And perhaps the AARP neurons are a locus of Sirtuin-1 mediated diaventomic effect connecting low energy availability and the immune system. Now, that diaventomic effect, that's all about what I'm trying to talk about here. So let's do a dialectic. The authors conclude by arguing in favor for the CNS involvement on the immune network, which can supervene leptin. I think we were pretty clear that's what the evidence shows. Perhaps the feeding stimulus itself involves an adaptive immune response. Adaptive immune response, you see? Conversely, now this is my caveat to this, it might be argued that the immunoepigenetic network connects the neural to the physiological response, and endocrine mediation runs parallel. So I'm turning it on its head and saying maybe it's the immunoepigenetic phenomenon that really helps regulate this throughout the feed-fast cycle. Now, why would that be preferable to having the feed-fast cycle regulate the peripheral immunity? Because the peripheral immunity is always surveilling, and it always has a complete network of activity from the brain throughout the entire aspect of the body, all the tissues. Right? So what would be a better mechanism than to use this continuous immune response? Right? And plus, the immune response is constantly detecting changes, which you would need to have to be able to inform the brain what's happening. Now, experiments that impose a specific T cell response, for example, Th17, okay, which we talked about a little bit in this paper. This is the idea I have. Could retrograde, like going back, the neuronal epigenetic changes involved in the endocrine system, such as a Sirtuin lack of expression. So, guess what? Here's a paper that was just published April 11th, so that was just a little over a week ago. In fact, it's still ahead of print, so you can't even get it in regular immunology journal yet, but it's published in Immunology, which is a darn good journal. Look at the title of this. The impacts of the microbiome metabolites on immune regulation and autoimmunity. Yeah, I'm not a big fan of the microbiome literature. There's a lot of hubris and hyperbole there. But I do look at it when it's specifically asking questions about the immune response, because those kinds of experiments are very, very precise and well controlled. And this particular paper did not make me feel otherwise, right? It didn't contradict the way I felt about that kind of research. So what did this paper say real quickly? Experimental autoimmune encephalomyelitis. We just looked back in the PNAS paper on this. Animal model of MS, as I said, suggests that modifying intestinal bacterial populations, microbiome story, influences the immune cell priming in the periphery, resulting in dysregulation of immune responses and neuroinflammatory processes of the CNS. This is the retrograde discussion I said was probably more likely what's happening. And here's the paper that is basically supporting that theory of mine. Okay. Conversely, some commensal bacteria and their antigenic products can protect against inflammation within the CNS. Okay, so obviously some bacteria are going to slow that process down. So bacterial induction of pro-inflammatory cytokines and subsequent generation of Th17 cells may be involved, because those are going to be major players here, Th17, those TFs. Commensal bacteria promote the generation of Tregs. Wow, full stop there. Remember what happened when we knocked out the Sirtuin? We lost Tregs, right? So, but when you have certain commensal bacteria, it means the ones that are supposed to be there, they promote Tregs, and that can contribute to overall immune suppression. Well known what happens in the gut. Now, here's even something really cool, because I'm a lipid biochemist, and I really think this is cool. Short-chain fatty acids may induce Tregs either by Gprotein coupled receptors or the inhibition of Sirtuins. Yeah, so short-chain fatty acids have all kinds of potential pleiotropic allosteric roles, right? This is not something that's gone unnoticed to lipid biochemists. We find this often, okay? So that's just something to keep in mind. So because diet, short-chain fatty acids, for example, or that is the incomplete beta oxidation of long-chain fatty acids, but diet represents an environmental component of a crosstalk between the gut microbiome and the immune system. So there you got environment, the immune system, and here you're adding in the entire enteric response, which is, of course, a neuroendocrine immune system. Now, I'm reminding you now what I meant about the diaventome. This is something that was in my last talk. I'm just going to bring this up at the very end of this talk. You have the genome, which is in pink for the geneticists. You have environment, which is in green for the environmentalists. And then you have the immunoepigenome, which is sort of this really cool mosaic of the two colors. I know, isn't it cool? Now let's look at this. Neural networks are not substances, they are processes. That means they're in constant dynamic agentic flux. They become eventual in nature as opposed to substantial. So they're not substances, they're events. An event ontology that reaches across the neural network, across this neural network here, of genetic and environmental interaction is articulated and synthesized through the epigenetic modification of what's known classically as the immune system. That will create a responsive plastic and elastic memory, so one that leaves a mark and one that pulls the mark back off. That's plastic versus elastic. Capable of learning, ideation, imagination, understanding through time. So not only are we talking about this is the neural connection, but now I'm showing you how the periphery functions on it. Pattern of events appear as energetic fields rather than particles of matter, much like chemical bonds. That's why I turned this into a chemical bond structure, right? A trigonal plane. Now, central, individual, agentically interacting through the events, these are events, obtained across the inherited genome, the changing environment, such as diet, and the transcendental or above immunoepigenome through time. So this is overall what I'm describing in terms of the diaventology of the system, trying to put together all the dimensions that regulate, in the one we just looked at, how peripheral immune responses are mechanistically controlled by the brain and then back to the brain. Can control things like autoimmune diseases, can cause brain inflammation, but can also decrease appetite and control appetite when functioning on the positive end of things. Okay, this is just another example. So I'm going to end now. Remember that this is Vera of Med. That's me. I'm Dr. Dan Guerra. And hopefully you enjoyed today's talk. I know it had a lot of technical information, sometimes more than this may have been more than the usual, but it was necessary for us to go through that PNAS paper. Hopefully you got something out of that. And likewise, hopefully I was able to put together at the end what I'm trying to say about how there is this structure that is in the living system, in the human system, that is controlled by the epigenetic modification of the immune response, which leads to interactions in both directions that are both plastic and elastic with the central nervous system that can regulate things like thermal balance, dietary feeding, bioenergetics, as well as the immune response itself. And when you tweak that system too much, for example, by knocking out an epigenetic reprogramming of the arcuate nucleus, AARP neurons, which then are going to desuppress the POMC neurons, leading to a particular phenotype, you can actually induce brain inflammation. Right? So that means you have to be very wary about drugs that may impact one aspect of obesity, like controlling SIRT1 activity in the hypothalamus, while not considering what can happen to an autoimmune response. So everything is obviously connected. It's connected through this diaventome, as I suggested. So all I've got to say now is thank you very much for your undivided attention. And hopefully we'll see each other again real soon. Again, if you have any questions about this talk, please contact me directly via my email, and we can discuss this and any other things you might be interested in the biomedical literature or the general biochemistry, cell physiology, pharmaceutical literature, to name a few. So the only thing I have to say now is bye for now. So I will say bye for now.